We do have Ian's presentation up and running. We're going to save that for the last presentation of the morning session. Ian's not here to answer questions, so we're going to try to make up our time and get out to lunch on time. Thank you.
Thanks, Corey. Hi, everyone. My name is Nadia Mamuzadeh, and I am going to be sharing with you a little bit more about the work that we are doing in Mariah Meeks Lab at Michigan State University. And here I'm going to be talking about our work to assess genetic diversity of brook trout across the Lake Superior Basin. But before I move any further along, I just wanted to acknowledge my collaborators on this work who are listed here, but also the many folks from many different agencies and organizations reflected by the logos here who have contributed to this research in one way or another. And so genetics is becoming an increasingly valuable tool for fisheries management, as we have heard already, and can provide information to guide and inform decision-making in a management context, including to support the FCO for brook trout in the Lake Superior Basin. And there are many types of information we can derive from genetics, but just to list some examples here, we can use genetic information to identify reproductively distinct populations, and this allows managers to then tailor management actions at the population level. We can also determine population health, and this is useful for prioritizing populations for management intervention. We can infer movement patterns to help understand spatial connectivity and even spatial habitat use. We can, as we heard from Ben, identify source populations in particular contexts, including for coastal brook trout. We can also evaluate hatchery contributions and gauge whether hatchery programs are achieving the goals that we intend them to achieve. And really what we're doing is we're living in a time where genetic information can, in some instances, be generated more cost-effectively to help meet these questions than it would take to collect the biological and ecological data to evaluate some of these questions individually. And as Chris gave a very nice overview of, there has been a fairly long history of studies aimed at evaluating genetic diversity in brook trout in the Lake Superior Basin. However, so far, many of these studies have been either primarily local or maybe even regional in their spatial extent, with a few notable exceptions, as we heard from Chris. And in addition to that, they have largely surveyed different sets of genetic markers, and this can be challenging because it means you can't combine data across studies. So while these studies have provided very important information for informing management, there really has been this need to develop basin-wide information using a standardized set of genetic markers so that we can continue to develop more comprehensive insights over time. And so this is the work that we are doing in the Meeks Lab at Michigan State. And for the Lake Superior Basin, this has kind of manifested itself in three different kind of thematic areas, three different studies, one of which we have already heard about from Ben. I have listed these in chronological order here. So what I'm going to focus on are two additional efforts, and I'll start with this one here, which is a study that we've already completed, which was focused on developing a SNP panel for brook trout in the Lake Superior Basin. So the specific objectives of this work were to, as I just said, create a SNP panel that can be used to survey genome-wide SNPs in Lake Superior Basin brook trout. And I'll talk a little bit more about the significance of this in a few slides, but really this gets at that data standardization piece. How do we quickly and effectively generate genomic data sets using similar sets of markers so we can expand on those data over time? And then the second goal of this study was to take that SNP panel and then use it to genotype brook trout across the basin so that we could start to develop baseline information on important population level relationships, including many of the relationships I highlighted on that previous slide. And what we found while we were exploring these objectives is that we could also start to pull out areas, geographically speaking, of uncertainty, which is equally important information for prioritizing future efforts. So the field sampling that went into this SNP panel design study is reflected in this map here. Each red point is a different site where brook trout were sampled in this effort. For this particular study, a lot of that sampling effort was led by the Ashland Fish and Wildlife Office in Wisconsin. However, we had lots of contributions in terms of samples as well as information to guide sampling from the partner agencies shown here, state, federal, tribal. And so this culminated in the sampling design shown here. At the end of the day, these sites corresponded with 74 rivers, 6 lake locations, and in addition to that, we surveyed 9 known hatchery strains previously or currently stocked in the Lake Superior Basin. So next I'm going to get into some results. What did we find? Well, first of all, we developed our SNP panel, and this SNP panel targets over 33,000 SNPs in the brook trout genome. And just to put that in some context, when we were working with microsatellites, it's often the case that you're surveying tens of regions of the genome. And with this change in technology and capabilities, we're now able to survey thousands of regions of the genome, including through use of this SNP panel. Why does this matter? 2 main reasons. First of all, we now have an improved workflow. It's more cost-effective and time-effective to develop genomic data sets for brook trout in Lake Superior now with this panel. And in addition to that, we can now very easily standardize our data by using this panel or complementary methods so that we can directly integrate data across studies. Moving into our next result, as I mentioned, we then took this panel and we used it to genotype many more brook trout from, again, the locations shown here. So in the end, we were able to survey genetic diversity in over 2,500 brook trout. This is the largest genetic data set for brook trout in the Great Lakes here in Lake Superior, which is really fantastic. And of those hatchery strains I mentioned earlier, we also genotyped over 100 fish. So what did we learn from those data? I'm going to share a little bit about our results that were focused, our analyses that were focused on identifying genetically distinct populations. And so this map looks very similar to some of the maps that Ben shared in his talk. At each sampling site, I have a different pie, and those pie slices are color-coded by genetic population. And before I go into any more detail about this, I just wanted to note here I'm using the Keweenaw Peninsula as an example, so about midway across the Michigan Upper Peninsula. But we have this information now for many locations across the Lake Superior Basin. Just a few additional examples, which I'm sharing with you here, which is really, really exciting. Okay, so back to the Keweenaw Peninsula, just as an example. So maybe one thing that might stand out to you is that every stream or river has its own genetic population tied to it. So again, we're seeing this really fine-scale population structure. And in addition to that, you might notice that there are some slices taken out of each pie, which reflect movement from other genetic populations of fish that were then sampled in a different location in this study. So just to highlight some of that movement, I'm going to move into a different type of result here, focused on this theme here. And I'm just going to use one example. Here, focusing on the Pilgrim River, which that genetic population is shown in this light blue. And actually, we recaptured some of those individuals from that population in a neighboring creek, Schlott Creek, which indicates that there must have been movement of brook trout through the Keweenaw waterway to that neighboring creek. Looking at this from the perspective of Schlott Creek, which again supported its own genetic population, shown here in the pink now, we also detected members from that pink population in the Pilgrim River, and similarly, some individuals in the nearby Gratiot River, reflecting movement into the lake and then sampled in the Gratiot River in this study. So we're starting to fill in some information about movements from these genetic data as well. And then the last result that I wanted to share from this particular study is really focused on our analyses to compare our known hatchery sampled individuals with our known wild sampled brook trout. And in this figure here, I am showing you the results of principal component analysis. And you're seeing axes 1 and 2 here. And I'm sure you can't make out the percentage of variation explained on each axis. But just to say, the main point of this type of analysis is just to get a sense of the overall genetic patterns reflected in the data, so overall patterns here. So each point is an individual brook trout. The circles are wild-caught fish, and they're color-coded by sampling location. And each triangle is a known hatchery origin fish, and those are colored by known hatchery strain. So again, we can start to get a sense of the relationship of our known hatchery fish, here are our coaster strains here, with our wild-caught fish. And some key takeaways that we had from these types of analyses were, first of all, we did not see evidence for widespread introgression of domestic broodstock in our wild populations. However, what we did find were a handful of previously stocked hatchery fish that were captured in our wild sampling efforts. Okay, so just wrapping up this study and kind of reiterating some key takeaways from this work. First of all, we now have a SNP panel that can be used in additional studies to survey genome-wide SNPs for brook trout in the Lake Superior Basin. In addition to that, one of the main things that we found is the presence of genetically distinct populations at very small spatial scales. By and large, and Ben described this too, we found genetic populations corresponding with every stream and river. And for large river basins with widely branching rivers, we were able to detect genetic populations corresponding with tributaries within rivers. So very fine-scale genetic diversity. In addition to that, as I mentioned in our analyses comparing hatchery and wild fish, we found a lack of widespread domestic introgression in wild populations, despite our ability to better resolve that introgression if it is there with our use of SNPs. And this is consistent with work that has been done prior to this study, and suggests that perhaps stocked fish aren't contributing meaningfully to wild recruitment. Additional insights, as I mentioned, kind of these basic population level relationships, some that I didn't have time to talk about today, but insights into population health as well as movement patterns, we saw an example there, and potential coaster source populations. Okay, so with that, I'm going to move into the second study that I'm going to share about today. And we're going to switch gears, and this study is focused on evaluating the potential for genomic impacts of splake on native trout in Lake Superior. And so just for everyone in the room that is maybe a little less familiar, splake are an anthropogenic hybrid of brook trout and lake trout that have been stocked in Lake Superior in some locations to help support important recreational angling opportunities. And our goal has been to evaluate what the genetic relationship is of splake to native populations of brook trout and lake trout. However, here I'm going to focus on brook trout given the brook trout theme of my talk. So the three primary aims of this work were first of all to determine whether splake introgression into wild populations of brook trout is occurring. And if it is occurring, to what extent is this happening? Can we pick out spatial areas that are more important than others? And this particular aim is really centered on addressing an information need identified by the Lake Committee and charged to the Lake Superior Tech Committee in 2018. But we have also added some complementary aims to this work. The second aim here is focused on using a hatchery experiment with known crosses of brook trout, lake trout, and splake, so known levels of introgression, so that we can characterize any potential fitness impacts that might correspond with a certain level of introgression. And with that aim, I'm going to take a second and just pause and describe what I mean by introgression. And really what all I mean there is the transfer of genetic material, DNA, from, in this case, one species to another species, which can be really problematic for the recipient species. And then the third aim of this work is to really take all of the data we are generating under this first aim here and leverage it in a different way. We have those data already, right? How can we use that to expand our baseline population level information for brook trout and many of the relationships I highlighted in that initial study? So where are we at in this project? This is currently an ongoing effort. It started about 2 years ago in March 2023. And since kicking off this work, much of our focus has been on collecting the samples through our many partner agencies to make this work a go. And our target was to sample 2,500 brook trout just for this effort alone. And we received the last samples for this effort to the MEEC lab in December of this past year. So during that time, we've been generating our data. Now we have moved into a new phase of the project. We are solely focused on processing those final samples that have come in. And soon we will be kicking off our third phase of the project, which will be focused on data analysis. So the main result that I wanted to share with you today is really what our sampling looks like now that we've completed that massive field sampling effort. And I say we, but really what I mean are the many partners that chipped in to make this project a go. And again, without this help, this project just simply wouldn't have happened. 2,500 brook trout is a large number to achieve for this species. So a thanks to our valued state, tribal, federal partners. We had an NGO and even another academic lab chipping in samples as well. So these locations here reflect the locations where new brook trout were newly sampled for this particular effort. And we met our 2,500 target sample count, and these reflect 74 rivers and streams across the basin. And again, the beauty of these genomic methods and being able to now combine our data sets is that we can now take the data that Ben described, including our initial effort through Minnesota DNR and our second effort, which Ben shared about, and the first study that I talked about in this study, and in this talk. And we can take all of these data together and now use them to evaluate our aims in this particular effort. So I think this is a really wonderful reflection and example of our capabilities now with genomic methods being online. Okay, and so just to put some numbers on it, we have data for over 6,000 brook trout, which is really fantastic, from 143 rivers and streams across the basin. So as I mentioned, this is an ongoing effort. We've kind of been moving through these different phases of the project. Our first phase was very field-focused, partner-heavy. We plan to continue engaging with partners as we work through our data generation stage, which we aim to wrap up this spring, and then spend lots of time focusing on lots of data that are being generated in this study. And so what is coming up next that we hope to share in future tech committee and hopefully late committee meetings as well are information insights that we are able to generate that are focused on these things here, which really speak to our main project aims. Determining whether slake introgression into wild populations of brook trout is even happening, and if so, what does that look like? And then second, continuing to build on our understanding and knowledge of these baseline population level relationships, refine our understanding of where genetic populations are located spatially, exploring movement patterns, hatchery contributions, all of these themes that we hope to be helpful and important for managers making decisions about brook trout in their various jurisdictions. Okay, so that's all I have. Again, I just wanted to thank, I'm here presenting on this, but really many people have contributed, as I mentioned, to this work in one way or another, especially through providing samples to make this work happen in the first place. And shout out to Mariah, who is also here at the meeting today with our MSU team. And I'm happy to take any questions in the time I have left.